developing fundamental unit operations that enabling self-driving labs. And I am cross-appointed to the Acceleration Consortium, so I'll show you a lot of the cool stuff that we are doing and the AC is working on. And together, the theme here is looking at how we can, using robotics or applied robotic science, to help us engineer frameworks that can help us to formulate and prolong viability of mitochondria. So starting with self-driving labs, I will start with this. Well, just show of hand, how many of you have sat in a self-driving car? Tesla, well, I see a few hands, Tesla. How many of you sat in a car that has adaptive cruise control? Yeah, see, that's not a full self-driving car, but it's a level 2. So you actually have a lot closer to think about self-driving cars than thinking about actual self-driving. But what I wanted to talk about here is how we're linking the concept of self-driving in the context of doing science. We're doing wet chemistry science. Self-driving cars are able to help you navigate and take care of some of the smaller tasks. For example, following the speed or be able to watch for objects on the road. But it's not going to tell you where you want to go. And same thing as doing science, there's a lot of fundamentals about how we can enabling robotics and autonomous workflows to help us. But ultimately, it is us. We're in the driver's seat. And by the way, don't ever do that, okay? It's really important. So when I was doing learning how to think about self-driving labs, a year ago, I did an experiment. So I went out to ChatGPT, and I said, I am a formulator, so I do formulations, make cocktails, and I'm using self-driving lab. Now, show me a picture of me. What does my life look like? And this is what ChatGPT did. So in a way, when you're thinking about self-driving labs, you're thinking, okay, well, there's a bunch of robots that are doing formulations and solution handling and testing for you, and meanwhile, you can feel sound asleep at home. That's really not correct, just like how we're talking about self-driving cars on the road. It's really about bringing us together using fundamental operations that enable us to make better decisions. So it's not about the machine will take care of everything. The machine will take care of things that will help us improve that precision. So this is an idea that I feel I wanted to drill in. Is that, this is building self-driving labs sounds really exciting, and it sounds new, but this concept has been around for a long time. Industry has been doing automation to improve speed on processing and analyticals for decades. And what's new is the newer emerging science on bringing visualizations and analytics to the powering the automation. But at the end of the day, none of this really makes any sense whatsoever without our human intuition. And so for a lot of the trainees in the room, if you're using ChatGPT or Gemini to help you write or help you assist you on doing various tasks, just remember that it's only able to do some to a certain extent. And in the case of talking about the self-driving labs, these tools can help us build patterns and see patterns, but ultimately, without us, those are just patterns. The meaning actually comes from us. So what we do is thinking about how we can combine these tools together and started to think about building a loop. So building an experiment, designing an experiment, building it, testing it, getting the results, and then trying to understand if we're on track or not. And then if we're not, how do we do it again? So you continue to regurgitating the loop. And so what's really exciting about self-driving lab, the power of it is the ability to do iteration. So that is one myth that I hope that a lot of the trainees are able to take home, is that it's not here to automate everything. But in fact, I would say that self-driving lab is only good and suitable for certain tasks, not all. And the tasks that are very, very good at are the tasks that are repetitive and are very short. So if your timeline for that experimentation is short, that is something that's very suitable for doing automation. If something that takes a long time to do, then you may not even get the benefit by putting all the work towards automation. Also, just like Melissa, I'm also cross-appointed to the acceleration consortium here, and I just want to bring it to the audience here, especially for those who are doing fundamental in biology and biochemistry. The self-driving lab, the acceleration consortium is a group of dedicated staff scientists that are here full-time, professional staff scientists that are working together to solve common problems. And these scientists are broken into individual teams. So they're SDL1 all the way to 7. They dedicated to these discoveries and autonomous loops on various tasks. So you have one for inorganic materials, organic, polymer, formulation. So I'm part of the advisor for formulation. Melissa is on the human organ mimicry. And really think of this as an infrastructure, both in terms of the equipment, but also in terms of the staff scientist expertise. So if there's some ways that we can accelerate biology or biochemistry discovery, this is where you can see a lot of integration that could help. Now, I wanted to say a few words about what I do and how we are applying the robotic workflows to accelerate for mitochondrial formulation discovery. So first, I wanted to show you a workflow that we've developed a year ago. This robot is called the RAISE robot. And RAISE here is a autonomous image surface evaluator. And what it does is able to use computer vision, so camera, combined with liquid handling to track a certain event and then be able to analyze that. And the very first thing that RAISE was doing was looking at the surface wetting properties. So this is a liquid handling robot, but it's no different than a pipette that you're handling powered by your thumb. And what we're doing is being able to not only track the dispensing of the fluid, but also using computer imaging to help us predict and measure the surface curvatures and angles, and that help us to determining the surface wettability. So Sheldon in the audience here is one of the original inventors and brought this robot to reality. And so by dragging these liquid, we can start to analyze the surface properties. So you might wonder, what does this have to do with mitochondria? Well, if you think about dispensing liquid, coating liquid, or putting a drop of mitochondrial formulation onto a substrate, the degree of surface wetting and then the contact adhesion and the uniformity of coating can actually all be captured and analyzed through this type of analysis. The other thing that we have done is looking at using imaging to track on evaporation behaviors. So you can look at how a drop of fluid dries, and the function of drying can be plotted against the residues, so you can see what is being left behind. And the rate of drying also teach you about the component mixtures inside. And of course, if we combine that with high-speed imaging, we're able to look at how the formulation spread on different surfaces. So this is a drop of fluid dropping onto a silicone surface. So it's a semi-permeable material. And by looking at the pattern, you can start to analyze on the cohesion force of the formulation. And it also tells you about the strength and the characteristics about how this material is contacted to the surface. So in a way, you can think of this is how we are documenting, digitizing that initial contact phase where a drop of biological fluid is contacting the surface. Things that we often take it for granted. And in fact, there's a lot of controls that you can do by further tuning here inside. So with that, we have built a robot that does autonomous driving. So you can think of a way of looking at this as a person designing the experiment and then executing it on a platform. And then the computer will essentially stop when it gives you the right answer. So see, you have to set the goals. It's not about just running the robot as just telling them to go explore, but saying, here are my objectives, and don't stop until you find these answers for me. So in this particular example, we're asking the robot to use a different a group of cocktails, and we tell them, go make me a cocktail that has a very specific wetting properties on the surface, and give us as many choices as you can. And we can also give you constraints, for example, on the cost. We can talk about the constraint on what is the maximum number of ingredients you can have. So this gives a lot of flexibilities in terms of the power for automation. But the other thing that I find was particularly useful here is the repeatability and reproducibility. If I ask someone to do surface wettability, so essentially pipetting a drop onto a surface and look at that surface property, if you keep doing that, for human, we get fatigue, and the error started getting bigger in terms of the consistency. But the robot is just doing the same thing over and over again. That's actually what it likes to do. So if you look at the plot on the lower left, the plot between the human operator versus a machine, you can see that, obviously there's a degree of accuracy that it has to be able to measure what we are measuring. But the error bar started getting smaller as we started to run this study over and over again, because obviously there's a repeatability errors that we tend to, it's actually quite difficult for us to overcome. So that's one example on surface wettability campaign. In the last year or so, we have built quite a few other adapting modules to it. So not only now is able to measure surface wettability, it's now able to do coating, is to do different kind of coating, spin coating, blade coating, and working with a colleague in mechanical engineering, Kevin Golleman, we started to look at how we can use the same design to test the surface properties. So we can do surface coating, we can do drying, we can also do scratching, just using our fingers to mimic that motion of scratching on a glass surface. So this type of unique workflows, it's quite powerful because it allows us to do a lot more focused time on designing the experiment as opposed to doing these repeating workflows over time. So these are some of the examples that I wanted to show you. And we have quite a few different robotic workflows that goes anywhere from testing the rheology functions all the way to measuring specific properties of a material surface in interfacial tensions. So with that, I wanted to give a quick overview on how we're using this to enable the mitochondrial transplant. And I really appreciate Anna bringing me on board because when she approached me, this is probably 4 years ago, talking about mitochondria, I hadn't thought that was an object in the cell. I did not think about it as alive. And so part of this central grant that we are working on, this NFRF team, is looking at prolonging the viability of mitochondria. And we had a lot of ideas on how to do so. Think about using coating, using different ingredients, or salt, or temperature conditions. And then these are, fairly complex set of parameters, and it's very difficult to actually do it. But here are the co-PIs and the collaborators that we have on this grant, and it's a huge effort. So the first thing I wanted to show you that we are doing is an underlying theme that for here is the very first task that I asked my team to think about is how can we quantify mitochondria viability as a function of time? And the simple answer is, can we actually help this group consortium set a benchmark to say, can we know how much mitochondria in a given batch or a given dose is alive and for how long? And how long can we track it? And that was the initial goal that we set out to do, and it was quite challenging. But we have some pretty exciting results to share. This is done by the postdoc in my lab, Sarah. She has been able to use fluorescent to help us identifying a way to track the membrane potential of mitochondria over a long period of time. To the best of our knowledge, I think most of the tracking is done within the first hour or 2 hours, but we have been able to do it for almost 7 hours, and the longest one that she has done is close to 10 hours to be able to track the membrane potentials. So this is quite important for us because fluorescent microscopy is great, but it's very difficult to autonomize. We're develop something that's a higher throughput, shorten the iteration cycle, if we're purely based on our quality of output, based on microscopy results. So this work shows that we're now able to track a mitochondrial membrane potential over the course of 4 hours, and it shows us the period of the dye equilibrium in a loading phase, followed by the plateau, and then a gradual decline. So this is going to give us the initial benchmark on understanding how the dye performs. The other thing that we started to do is to start building a larger framework on educating ourselves and then building the model to autonomize the workflow. And do this, we have started building a machine learning foundational framework, and in collaboration with Mohammed Musavi here, who's going to speak afterwards too. And we started to curate all the papers out there that are able to digest. But it turns out there are only 32 papers in the literature to date that focus on material in mitochondria. And out of that, only 4 of the paper that actually tracks the viabilities. So there's not a lot of data out there, and this gives us a bit of a room for thinking about how we can do this on a larger scale. But what I want to show here is the graph on the middle here shows is the type of materials that were mined to do mitochondrial coding. But if you take a step back and looking at live cells that are much higher frequency of literatures, you can see that there's mostly using different materials. I think this gives us a much better opportunity to open up the design space. And so ultimately, what we're going to do is to build a network that can develop a workflow that trains not only on the preparation, but also doing the functionalization and the data analysis into the network. Notice I'm over time, so I'll stop here, and I'll thank my team. And most importantly, I want to thank the NFRF and this wonderful network led by Anna. Thank you.